Hello, ladies and gentlemen. Today we are going to be talking about viruses. All the shapes and sizes. Viruses. So, are they dead or are they alive? Let's check it out. Living things consist of 7 characteristics of life. Made of cells, metabolism, respond to environmental stimuli, grow and develop, contain DNA or RNA, reproduce, and maintain homeostasis. Let's see how viruses add up. First off, they do have a capsule that goes around their genetic material, so that would count as a cell. Okay. Next, they evolve, mutate, and change, so they respond to environmental stimuli. Yes, they contain DNA or RNA. That's their genetic material. Now, reproduction. Reproduce. It's sort of iffy. They can't do it on their own. They must actually be inside of a host and use the host machinery to make more of them. So they can't do it on their own, but they get the job done. So, Next, maintain homeostasis. We see that capsule again, and it does keep the insides away from the outsides. So again, that's sort of an So, well, we got 4 out of 7. Some scientists say yes, some scientists say no. This is still actually a big debate right now. It depends on how many characteristics you use, depends on what definition of life you use. So you will need to make up your mind and make a judgment and be able to back your judgment with evidence. That would be a really, really good essay question. Hint, hint, wink, wink. Yeah. Okay, viral structure. Start with the pieces. The viron, or the viron, either way, is the complete infectious particle. It's the whole package. Next we have the capsid. Capsid is the protein coat that encloses the genome, the DNA or RNA. That protein coat helps recognize host cells. Okay, the capsomere are the little particles that make up the capsid. The nucleic acid, genetic material, the DNA or the RNA, makes sense. Now, nucleocapsid, exactly what it sounds like. The combination of the genetic material plus the capsid. Together, nucleocapsid. There we go. Some have an envelope. Not all, but some have an envelope, which is a protein membrane that encloses the nucleocapsid. It is protein and glycoprotein. Now let's take a look at some shapes. First we have the helical virus, which again has the capsid surrounding the nucleic acids or the genome. And we have the capsomere, which are each individual piece that makes up the capsid. Okay? Helical. Next we have the polyhedral, which means many sides of virus. Polyhedral, again we have the whole capsid made up of the capsomeres, and we've got the nucleic acids inside. Makes sense. Next, we have the enveloped virus. We've got the nucleocapsid inside, which is our capsid with our nucleic acid inside, made up of the capsomeres. And then we've got an envelope around it. Finally, we have our complex viruses, or our bacteriophages. We have the capsid as the head. We have the DNA inside. We've got a sheath. We've got the plate with some pins, and we've got the tail fibers. Now, the base pins and tail fibers help it actually land on the cell. Then it actually squeezes its DNA into the cell. Okay? We'll learn more about that when we talk about reproduction. This is going to be your hated most part. You'll need to draw these. Now, not in detail. You do not have to draw every single one of these capsomeres. Do not do that. I want shape and where the DNA is located, or the RNA, the genome. So give me the outline of this guy, and then do some squiggles inside and look about the nucleic acids. Give me some roundness, show me, the helix, and show me where the DNA is. Again, with this guy, the outside shape, Then we've got the capsid, and then we've got the nucleic acids inside. Again, do not draw me in detail. You can, but you don't have to. Same with the bacteriophage. Should be the capsid, should be a sheath, should be a base plate, and some legs coming off it. Looks like a lunar lander. Look at these in more detail. Helical virus, bigger picture. It is a long rod shape. The rod can be flexible or it can be rigid. It's got the viral genome in a hollow cylindrical capsid. And we've got two examples. This is the rabies virus over here. And you'll know about the Ebola virus, how it moves and loops. So the Ebola virus is an example of it being flexible. And the rabies virus here is an example of it being rigid. Polyhedral, just many-side. So most of the time we see an icosahedron, which is 20 sides with triangle faces. We have examples of an adrenovirus. We have our HIV up here with the many sides, and we also have our poliovirus. Next we have our enveloped virus. It's roughly spherical, not always, maybe more tube versus sphere. But there's two types. We can have an enveloped helical virus or an enveloped polyhedral virus. Basically, we took the other two and we put an envelope around them. So we've got the influenza that's over here. Again, we see our envelope around our helical capsid. And here we got an enveloped polyhedral. We've got our herpes simplex virus surrounded by the envelope. Yay! Complex viruses, bacteriophages, one of my favorites. The capsid is a polyhedral. Then we have the tail sheath is a helical type thing. We've got our tail fibers, and we've got our plate and pins. So again, we see this guy here, and he is actually literally gotten onto a cell, and he's injecting his DNA into the cell. So he doesn't even need to go into the cell. He just injects his DNA. Or RNA. We'll talk about that more later. So, sizes. They're measured in nanometers, 10 to the negative 9th. Please make a chart of each of these telling me their name and sizes. So, example. C4 bacteriophage, 225 nanometers. Adenovirus, 90 nanometers. You get the idea. Now guys, take a look over here at the human blood cell. It is 10,000 nanometers versus, all these tiny dudes, 30 nanometers. These viruses are teeny tiny, but they can do so much damage. Okay, so viral replication. They're hijacking or taking over our cells. There's one major pathway, and then we can have a detour. The lytic cycle is the major pathway. It goes 1, 2, 3, 4, 5. The virion, or the virus, attaches to the host cell. It then releases its genome into the cell. The genome is then replicated, so by the host machinery, and we use transcription and translation to make more of the viral proteins. We then take those viral genomes and all those proteins and assemble a new virus inside of our cells. And then we have to release the infectious virion to find a new host. Now that's the lytic cycle. That's the normal thing: attachment, entry, production, assembly, release. We can have a detour in there that we call the lysogenic cycle. And it happens right after the genome entry. And it's between steps 2 and 3, the lysogenic cycle. That DNA, that viral DNA, can actually incorporate or combine with our or the host DNA. Once it's combined into our genome, it just sits there and it goes dormant. It doesn't do anything. As our cells reproduce and copy our DNA to create more and more and more, every single cell that we create has that piece of viral genome inside of the DNA. At some point in time later on, there's a trigger that causes that viral genome or that viral piece of DNA to come out and to go ahead and hop back into the lytic cycle and be replicated and make viral proteins so that it can be assembled and then have more virus release. Okay, so again, there are two pathways. We have the lytic cycle and the lysogenic cycle. Let's take a look at one. So the virus must come in and attach and then enter. Once it's entered, it must release its genome. In this case, we have the viral DNA right here. It moves into the nucleus. It creates RNA to be transcribed and translated to make protein. And we also copy the viral DNA to make more viral genomes. Well, the viral genome comes out here, and it is released by the cell through endocytosis. We have all these viral proteins along the outside of the plasma membrane, and as it engulfs or envelopes the viral genome, it releases a new infectious virion to go out and find a new host. There's many ways to actually enter our cells. A virus can enter straight through. A virus can just inject the viral genome, which is down here in these bacteriophages. It has the genome in the top. It sits down, squats down, and injects it into the host cell. The virus can be brought in through endocytosis. Or the viral envelope binds to the host envelope, and as it widens, the viral genome is released into the host cell. That's about getting the DNA in. Once it's in, or the RNA, the viral genome can either be single-stranded or double-stranded. It can either be DNA or RNA. But whatever the genome is, it must make it from the cytoplasm to the host nucleus. The viral DNA is copied through replication, and it is also going through transcription and translation to create more viral protein. Or, remember, we could have that detour, the lytic cycle. The DNA can integrate into the host genome and lie dormant for a while, and then later on come back out and join back in the lytic cycle. Once the viral proteins are created, we have assembly by the host machinery. We take the genome and we pack it inside and we create new viruses. The virus release, or the virion release, the new infectious virions, which are the viruses, are released from the cell to go out and hijack a new cell. We can have two ways. We can either have lysis of the cell, which means it bursts and all the virions are released that are on the inside, or we can have the cell release the viruses one at a time through exocytosis. The virus is being enveloped by the modified plasma membrane. We'll see that in one moment. Here's an example of the host cell lysing, releasing all these bacteriophages. This actually was a bacteria, and you can see all of the heads of the viruses here. And as it bursts, it releases all the bacteriophages to go out and find a new host cell. Here's releasing by exocytosis. So all of these viral proteins that are created are put onto the outside portion of the cell. And then the viral genome is packaged as it goes out through exocytosis to create a new virion. This does not cause the cell to burst, and this cell will continue to make more and more and more viruses. Let's see if we can review. Well, we have the lytic cycle, which is 1, 2, 3, 4, and 5. And that's going to be attachment and entry. Then we have our detour, which is our lysogenic cycle, which the DNA can then integrate into the host cells. Or we can have that DNA be copied and replicated and go through transcription and translation to create viral proteins in step 3. 4 would be packaging and assembling. And then 5 would be release. Well, that's all I've got for you today. You are so getting hijacked. Bye!